today I would like to talk about the amino acid. Amino acids are the building blocks of proteins. Proteins are important of our body because they help with the growth and repair. Amino acids are special because they can act as both an acid and a base. They are changed depending on the pH, and pH tells us if something is acidic or basic. When pH changes, the shape and function of the proteins can also change. Now we proceed to the real world relevance. What is important? In our body, pH must stay stable. If pH change, protein like hemoglobin may not work properly. Second, in laboratories, scientists use pH to separate protein for medical tests. Next, lastly, in food production, like making cheese, milk proteins change when they reach a certain pH from curds. That's all from me.
There are several parts of key chemistry concept in this topic that we should know. The first part is of course nomenclature. For amino acid, they are named as amino derivative of carboxylic acid which the O with acid. These are some examples of IUPAC name and common name for amino acid. The next part is deuterium and isoelectric point. Deuterium is a molecule with an overall charge of 0, which NH3+ has positive charge and COO- has negative charge in the same molecules. In this diagram, if it is in low pH solution, it will act as a base and accept proton. This means COO- will accept H+ and turns into COOH and the overall charge is positive. Meanwhile, if it is in high pH solution, it will act as an acid and donates proton. This means NH3+ will lose H+ and turns into NH2 and the overall charge is negative. The difference of isoelectric point from deuterium is it is a pH which the amino acid exists predominantly as deuterium. Next, we will continue with the reaction of amino acid. There are 4 different reactions of amino acid. The first one is amino acid react with acid to form salt. Second is react with base to form salt and water. Third is amino acid react with alcohol in the presence of an acid catalyst to form ester and water. Lastly is reaction with nitrous acid. In this reaction, there are several steps until it forms the final product. They will react at low temperature to form diazonium salt. Then the diazonium salt decomposes to nitrogen gas and carbocation. Finally, carbocation react with any nucleophiles or undergo elimination reaction to form alkene. Now, we already reached the last part for key chemistry concept, which is the formation of peptide bonds. Peptide bond is formed from carboxyl group of one amino acid react with the amino group of another amino acid to form an amide by eliminating a water molecule. The amide is called a peptide, and the amide bond is called a peptide bond. Peptide may be formed from 2 or more amino acids. 2 amino acids react to form a peptide, which is called dipeptide.
Next, we will dive into a real-life application of charge of amino acids. First, let's look at medicine. pH plays an important role in how proteins behave. Amino acids in proteins change their charge depending on pH, and this affects how drugs bind to proteins. For example, enzymes in the stomach work in a very acidic environment around pH 2, while blood is slightly alkaline at pH 7.4. Because of this difference, drug effectiveness can change depending on where the drug acts in the body. pH is also useful in disease diagnosis. Changes in protein charge can indicate abnormalities such as unusual hemoglobin in blood tests. Techniques like electrophoresis separate proteins based on their charge, helping doctors detect certain diseases. Next, in daily life, pH affects protein around us. For skincare products, our skin has a natural pH of about 5.5. This helps maintain the skin's protective barrier. Using products with the wrong pH can disrupt protein charge and cause irritation or dryness. In food, protein behavior changes during cooking. For example, when we cook eggs, heat and pH changes cause protein to denature and become solid. In cheese making, pH changes cause proteins to coagulate and form curds. At the isoelectric point, amino acids have no net charge, so proteins may precipitate. Finally, in industry, controlling pH is very important. In food industry, pH is adjusted to improve texture in products like yogurt and cheese. In pharmaceutical production, isoelectric focusing is used to separate and purify proteins. In biotechnology, enzymes work best at their optimum pH, such as alkaline detergents where enzymes remain active. Overall, pH controls protein structure, solubility, function in medicine, daily life, and industry.
Today, I will be presenting part 4, group prediction, where we analyze how amino acids like glycine behave under biological conditions. First, let's look at glycine at a neutral pH. It exists as a deuterium carrying both a positive and negative charge. However, as it moves through the body, its environment changes its chemistry. In the highly acidic stomach pH 2, glycine accepts protons, giving it a net positive charge. In the near-neutral bloodstream pH 7.4, it remains as deuterium, but as pH rises, it begins to donate protons, shifting toward a net negative charge. Understanding this shift is the key to predicting how amino acids behave in different biological environments. Next, we'll explore about the logic and the limits. Predicting this charge is incredibly useful, particularly in electrophoresis for separating proteins, but there are trade-offs. Proteins are less soluble at their isoelectric point, which causes clumping. This is actually a benefit in food science for texturing. However, our predictions have limits. They often assume a pure water environment. In the real world, biological salts and complex 3D protein structures can shock amino acids from proton exchange. Looking ahead, this science is driving major innovations from three aspects. In health, we are developing pH-responsive coding for targeted drug delivery. In environment sustainability, understanding charge helps us more efficiently extract plant proteins for alternatives like soy milk. Finally, AI-driven enzyme engineering is allowing us to create stable proteins specifically designed for environmental cleanup, such as breaking down toxic waste and ocean pollution. By mastering molecular charge, we are building a more sustainable and healthier future.
We learned a lot about the charge of amino acid from the previous slide. So to sum up, there are three main points that we can take notes on. So the first one are the concept or the nomenclature, and the common name they use are leucine and serine. As for the deuterium, it's an equal concentration of anion and cation, and overall charge is 0. For the isoelectric point, the number of positive and negative charge of amino acid is equal, and the reaction of amino acid is composed of acid to form salts, base to form salts and water, alcohol in the presence of catalyst to form esters and water, nitrous acid to form diazonium salt. And the formation of peptide from the carboxyl group reacts with the amino group that creates amide. Amide non-peptide bonds are from 2 or more amino acids. So for the application, for the medicine, are used for the drug design and delivery, and for disease diagnosis, it's affect how drugs bind to the protein. And for the daily life, it's for the skincare product and food protein behavior, as it maintains a barrier function. For industry, it's used for food industry, pharmaceutical production, and biotechnology, as for the protein purification. And the lastly is analysis. It's about the glycine in our body, in location it contained in stomach and bloodstream. Stomach is the highly acidic environment, as the deuterium state, to carry a positive charge, where in the bloodstream it's near neutral and the deuterium switch to the negative charge. The advantages and limitation, however, advantages are the separation protein through electrophoresis and controlling solubility. Limitation are about the folded protein problem and prediction to be the less accurate. And for the trends and impact, it concludes of health, and the trend are developing drug delivery and impact design pill coating. Society position protein extraction and impact force them to separate from water. Environment are the trend of AI enzyme engineering, impact are clean up acidic waste. So that's all for the summary.